Hi everyone, and welcome to the Chapter 6 lecture for microbiology. This chapter is all about microbial nutrition and growth, which is kind of an extension or continuation of what we started to talk about in Chapter 2 when we talked about growing bacteria, like in culture media. We talked about different types of growth media. Well, now we're going to sort of zoom in on the actual nutrients that microbes need to grow and a little bit of the biochemistry involved in metabolism. So all organisms have essential nutrients. These are nutrients that they need to consume because their cells can't generate them. So we have essential nutrients as humans, animals have essential nutrients, bacteria have essential nutrients, and each organism has different essential nutrients based on their biological needs. All organisms need roughly the same chemicals to get by, just some have the ability to generate them while others do not. So we can roughly or broadly classify nutrients into two categories, the macronutrients and the micronutrients. So macro means large. These are nutrients we need in large quantities, and these tend to be organic biomolecules like carbs and proteins and fats, whereas micronutrients are things we need in smaller quantities or that bacteria need in smaller quantities or trace amounts. And these are oftentimes metals like iron and zinc. So yeah, and all this is just a picture down here of microbes that, all look very different. So it makes sense that they all have different needs. They have different enzymes, different biologies, and different nutrient needs. Another nutrient that's really important and sometimes overlooked, my husband just got home and the dog is going nuts downstairs. I don't know if you can hear that. And he got us dinner. So diffusion is the movement of particles from an area of high concentration to an area of low concentration. And osmosis is the flow or diffusion of water. Now, water is important for all organisms. It's hugely, 70% of Earth is covered in water. Our bodies are roughly 70% water. There are many environments that are aqueous, and pretty much all organisms need water to survive. The cells are full of aqueous solutions, and so water has the ability to move in and out of the cell freely across the cell membrane, which is selectively permeable. Right? So osmosis sometimes sounds like the opposite of diffusion, because osmosis is the movement of water from a low concentration to high concentration, as you can see in this animation on the right here. But that's because when we use the term concentration, we're referring to the amount of solute, not the amount of water. And so if we look at the concentration of water, the solution or the fluid on the left is the one that has more water in it than the fluid on the right. And so water is moving from an area of high water concentration to an area of low water concentration. Just the term concentration is not used that way. So osmosis is the diffusion of water from an area of high water molecules to low water molecules, but it's more commonly stated as the flow of water from a low concentration to a high concentration solution. A common way that nursing students in the past have told me that they remember it is they have been told that water follows solute. And it does. So you can think of water as being attracted to solute. So whichever side of the membrane there are more solute molecules, the water will try to flow to. Essentially what's happening is these two solutions are trying to equilibrate. They're trying to get to equal concentrations, but the sugar can't move. So the only way they can try to equilibrate is through the water moving. And if there was not the atmospheric pressure, and the weight of the water wasn't pushing down, then all of the water would move through. But eventually so much water piles up that the osmotic pressure, the pressure or weight of the water pushing down, kind of pushes against the water that's trying to move through. So eventually it does level off. and doesn't actually reach equal solutions or equal concentrations. But osmosis plays a big role, particularly for single-celled organisms. So for a cell, water is constantly moving in and out. And ideally, cells like to find themselves in environments where the osmotic pressure or the concentration outside of the cell is the same as inside of the cell. That's called an isotonic situation, or the cell is in an isotonic solution. Iso means same, so the concentration inside and outside the cell are the same. And when that happens, water moves in and out at equal rates, and so there's no like net gain of water, and the cell's kind of at its happy, homeostasis. If you put a cell in a hypotonic solution, hypo means less than. So hypotonic solutions have a lower concentration than inside the cell. And when that happens, water will flow towards solute, which in this case is inside the cell, higher inside the cell, and that will cause the cell to swell. And this is where we can see the real purpose of the bacterial cell wall and microbial cell walls, because the cell wall really helps the cell resist that osmotic pressure of the swelling when water flows in. All right, cells without a cell wall, they swell and rupture and burst. And you can see this actually under a microscope. If you do this with blood cells, you can like prick your finger and put some blood cells on a slide and look at it under the microscope, and then add some pure water to the slide, and all of the red blood cells just pop. They pop open because water is a hypotonic solution. Our bodies have a little bit of salt, sodium in the blood, and electrolytes in the blood, and so when we add pure water, that's why we don't give pure water via IV to people, because that would be putting their blood cells in a hypotonic solution, and it would cause blood cell lysis. So never inject pure water in someone. Always inject isotonic solutions. A hypertonic solution is the opposite. So hyper means excessive. So this is a solution that has a higher solute concentration outside the cell than inside the cell, and this draws water out of the cell and essentially dehydrates it and causes it to shrivel up. All right, cells need water to do chemical processes, so this dehydration can be fatal for a cell. Sometimes it can be reversible, but it's definitely inhibitory to growth. So we'll talk about in later chapters some ways that we preserve foods, for example, is by adding salt or sugar to them, because that creates a hypertonic environment that bacteria can't thrive in. When we are trying to get things in and out of our cells, osmosis is a form of what's called passive transport, so water and gases like oxygen and carbon dioxide, they can passively move and travel just straight through that phospholipid bilayer, no problem. All right, if you think of the phospholipid bilayer as like a fence, they're small enough to fit through those holes in the fence. But like if we're thinking about this fence, larger things like animals are not able to fit through the holes, and they need to go through an opening, a gate in the fence, right? So that's what these channels are for. There are protein channels that allow larger things to get through the membrane to either enter or exit the cell. The two types of channels are ones that are facilitated transport. So facilitated transport is another form of passive transport in the sense that these molecules are just diffusing into the cell through this channel. So it's kind of open, and molecules will only flow one way. And the way they will flow is from area of higher concentration to the area of lower concentration. They will naturally flow in that direction. Versus when you want to move something from an area of low concentration to an area of high concentration, that doesn't happen spontaneously. So if you want to concentrate some chemical somewhere, you have to use energy. I kind of think of diffusion and flow of nutrients as kind of like a river with a current. So if you are canoeing down a river and you're going with the flow of the current, you can like kick up your feet and put your paddle away and just float because the river's moving you spontaneously, right? It doesn't take any energy it takes very little energy to paddle downstream, all right? But then you turn around to go back, and then it takes a lot of energy to go against the stream, against the flow or current. And so you can kind of think of diffusion as a current, all these molecules going this way. And so if you're the molecule trying to go this way, it's going to be strenuous, and so it requires energy. So we call this active transport. It's active because it requires energy, and the energy that cells use is ATP. Some ways that cells get nutrients is through their membrane, through diffusion, or through transport. Right? But sometimes even larger things can be taken up into cells through this process of what you can think of as cell eating and cell drinking, phagocytosis and pinocytosis. Phagocytosis is pinching off of solid particulates, and pinocytosis is ingesting liquid components. So this image here kind of shows the two of them. Here's phagocytosis, a cell eating a solid particle versus eating some extracellular fluid with different, solutes dissolved in it. Both ways, they're just pinching off a piece of the cell membrane into a vesicle called a phagosome, or a food vacuole, and then oftentimes that fuses with a lysosome so that the contents get digested. So in addition to needing certain nutrients, bacteria also have several environmental factors that influence their growth. And the first one is temperature. So where do I want to put my face? Let's put it here. There we go. Okay, so all organisms have optimal growth temperatures, temperature ranges in which they can grow and survive. So bacteria are no different. Bacteria have a wide range though, and we can kind of classify and group bacteria based on what their optimal growth temperatures are. So I want to clarify that an optimal growth temperature is not a single number. It's a range. All right, so the optimum growth temperature is like somewhere across this top of the peak here, right? And then there's a minimum and a maximum, and temperatures outside of that, the bacteria really can't grow. So we can graph bacteria based on their growth temperatures, their optimal growth temperature ranges. And I'll start here on the left. So in blue here, these are microbes that grow at really cold temperatures, so like less than freezing, and their optimum is a little bit higher than freezing, so around 5 degrees Celsius. The psychrophiles are the ones that like it really cold, and the psychrotrophs like it kind of cold, so we call these, the psychro is referring to cold. In green, these mesophiles, notice that their optimum temperature is right around 37 degrees Celsius, which happens to be normal body temperature. So pathogens tend to fall into this mesophilic group. Thermophiles and hyperthermophiles, or extreme thermophiles, like hot temperatures. Thermo meaning temperature, meaning heat. And these are found in locations on Earth that are very warm. Another thing that's important for organisms is their gas intake. And the main gases that we care about are oxygen and carbon dioxide. So a lot of microbes use oxygen just like we do, but for some of them, oxygen is toxic. So oxygen, we're led to believe that it's like this life-giving gas, our cells need it, but it is also toxic because oxygen, which the chemical symbol for oxygen gas is O2, okay, it oxidizes things and it ends up with forming these toxic byproducts like peroxide ions and hydrogen peroxide and free radicals, and so those things actually damage cells. You may have heard the term oxidative stress or oxidative damage, and antioxidants being a good thing because they're basically things that protect us from these toxic byproducts of oxygen. And some microbes don't have those antioxidants, essentially. They don't have enzymes and molecules to detoxify oxygen byproducts, and so therefore the oxygen just kills them. So microbes they can fall into three categories. They can fall into ones that use oxygen, that need oxygen, and because they have the ability to detoxify it, like we do. Those that can't use oxygen and can't detoxify it, so in that case, oxygen is poison to them. And then there's a third group, ones that they don't use the oxygen, but they have a way to detoxify it so it's not toxic to them. So they kind of like don't care. They are apathetic to oxygen, like, take it or leave it. And we'll talk about the names of these different things. So cells that can tolerate oxygen must have the ability to detoxify it, and the two enzymes that are really important for detoxifying these byproducts, harmful byproducts of oxygen, are superoxide dismutase and catalase. And catalase breaks down hydrogen peroxide into water and oxygen gas, and you can see this like in you can do like a lab at home if you want to just get some hydrogen peroxide, and you can add it to potato, or you can add some yeast. Both of those things contain catalase, and you'll get bubbles. You'll see these bubbles forming of the oxygen. And so you can see that catalase in action. But these two enzymes are particularly important for being able to deal with and handle oxygen, specifically the oxygen byproducts that are toxic. So this is a nice table from your textbook that shows different levels of oxygen tolerance, I suppose, in microbes. And the three that I want you to know are the aerobes, the facultative anaerobes, and the anaerobes. So sort of the three categories I mentioned on the previous slide. So the aerobes, these are the ones that need oxygen, they can detoxify it, and they need it. They use oxygen like we do. They breathe oxygen. And so if you were to use this sort of what's called like a semi-solid media, and you stab the bacteria down in here, you'll see they only grow at the top. These aerobes will only grow at the top where there's plenty of oxygen. All right, as there's less and less oxygen as you go down here in this media, they can't survive. Facultative anaerobes, next I'm going to do anaerobes. Anaerobe means without air. And these are the microbes that can't detoxify oxygen. So oxygen is essentially toxic to them. So if you do a stab tube here and you stab bacteria into this media, you'll see that they grow at the bottom where there's no oxygen, and at the top they don't grow because the oxygen is toxic to them and kills them. Facultative anaerobes are ones that can, they don't need oxygen, they don't use it necessarily for metabolism, but they have the ability to detoxify it. So they can grow with or without oxygen, it doesn't matter. You'll see them grow throughout the media in the low oxygen areas and in the high oxygen areas. Like they just don't care. The oxygen's like, doesn't matter whether oxygen's there or not, they can grow either way. Another environmental factor that's important for growth is pH. pH or acids and bases can be denaturing agents. They can cause proteins to unfold. And so proteins are really, they're designed for a certain pH. So there are bacteria that can survive in acidic environments, and there are bacteria that can survive in basic or alkaline environments. We call them acidophiles and alkalinophiles. And they don't like neutral pHs. So kind of like with temperature, there are microbes that can survive in pretty much all the different pH ranges. So whichever pH range they're adapted to, that is the optimum pH for them. Most organisms grow best at a pH of 6 to 8 because most environments fall within that pH range. And also acids and bases can denature proteins. So some examples of some microbes that grow at different pHs. So Helicobacter pylori is a bacteria that causes stomach ulcers, and it's very happy living in the stomach, which has a very acidic pH, a low pH of 1.5 to 3.5, so roughly 2. Lactobacillus is an acidophile, and it's the one that grows in yogurt. It turns milk into yogurt by basically producing acid, because it likes an acidic environment. It produces acid and it makes its environment acidic, and that acid is what curdles the milk and makes the yogurt. Proteus mirabilis likes a slightly basic environment and actually makes the environment basic. It can cause UTIs. And so your normal pH of the urine is actually pretty neutral. But if you have an alkaline pH, so slightly basic, that can be a sign of a urinary tract infection. Just so you know, just to remind you, that low pH is acidic and high pH is basic. And my favorite mnemonic to remember that is, she's a 10, maybe on the pH scale, because she's basic. That was me trying to give my sass attitude, but. All right, osmotic pressure is the fourth thing that's important to microbes. So the concentration of their environment, essentially. We already went over this, the consequences of being in different concentration environments than exist inside the cell. So all organisms want to be in an isotonic solution, one that's equal to the concentration, that the concentration in the environment is equal to the concentration inside the cell. Some cells really like salty environments. We call them halophiles. Halo meaning salty. One example of a halophile is sort of a mild halophile, doesn't mind a little bit of salt, are a lot of staphylococcal species. And staphylococcal species, you find a lot of them on human skin. And human skin is a salty environment because think of sweating. When we sweat, we excrete salts. And so human skin is actually a little bit of a salty environment. And staphylococci, so organisms that live on the skin, do have to be halophiles or facultative halophiles, meaning they can tolerate at least. They don't mind a bunch of salt. They don't necessarily need it, but they don't mind it. Some other things that microbes are sensitive to in their environment are radiation and atmospheric pressure. So certain types of radiation are damaging to cells, like UV radiation and X-rays. That's why we wear protection when we're exposed to those types of radiation. We wear sunscreen to protect from UV burns, and we wear like a lead apron when we're getting an X-ray to protect our really ovaries and testes and reproductive cells from DNA damage. So since microbes, we have, several layers of tissue, and if some of our cell, like if you get an X-ray of your arm, all right, there are some cells in your arm that will sustain DNA damage and then they'll just die. But you have so many trillions of cells in your arm, it doesn't matter if a couple of them die. Versus, a bacteria that is one cell. Yeah, so I suppose if they grow in thick, colonies, then the cells on the bottom of the colony are resistant because the cells on the top absorb all those rays, but they don't tend to grow that thickly in nature. So that's why we use radiation as a sterilizing method. So it's used in the food industry. We irradiate some foods to kill any bacteria that might be on the surface. And products, some products are sterilized that way, like plastics. And we also use UV to sterilize surfaces. You can buy, in the pandemic, UV lights were something that were used to help sterilize because, again, it breaks DNA. So viruses have DNA, and so UV light can be used to sterilize by breaking DNA. Barophiles, baro as in barometric pressure, is a measure of atmospheric pressure. So barophiles are ones that are adapted to living in high pressure environments, like deep, deep sea, where the pressure is very high. So microbes, we've talked a little bit about quorum sensing and how microbes can kind of form these communities. They can also form communities with other organisms, like the fact that we have an entire microbiome, a collection of bacteria that outnumber us, that live on our body. And we've talked about fungi that live on the roots of plants and help to provide it with nutrients. There's all these relationships that microbes can have with other organisms. And so these different relationships ecologically are classified as follows. So you have this symbiotic relationships. These are ones where they live close together and benefit each other. They don't necessarily benefit each other, sorry. They live close together. And they have a relationship with each other. So it could either be that it's a relationship where both parties benefit. So presumably we benefit from some of our gut microbes that help to digest certain nutrients for us, and they benefit from us because we feed them and provide them safe shelter. Commensalism is where one partner benefits but the other member does not. And you still will hear people talk about our commensal bacteria and commensal microbes because for a long time people thought that our microbes were freeloaders, like that the gut microbes, that they didn't actually do anything for us. They just, they were just squatters and benefiting from our shelter and our nutrients. But in recent years, we've realized it's actually more of a mutualistic relationship. And then there's parasitism. So parasitism would be sort of a negative symbiotic relationship where one organism benefits while the other suffers or is harmed in some way. Non-symbiotic relationships include synergism and antagonism. So synergism is working together. So two different organisms that work together. They don't necessarily one doesn't live on the other, but they live in the same community and they work towards a common goal. So kind of like biofilm formation is a form of synergism when bacteria have that interspecies conversation through molecules. They can synergize. Antagonism is also a really important ecological relationship. So this is how microbes kind of fend each other off and protect their territory. So fungi that make antibiotics, all right, that's an antagonistic relationship that they have with bacteria around them. And antagonism actually can work to our benefit in many ways, health-wise. Because most antibiotics on the market are actually derived from bacteria or fungi that naturally generate them as in an antagonistic type of relationship. And also our microbiome they're sort of like our bodyguards. We are their turf and they defend their turf against pathogens or invaders. So there is a bit of antagonism between our normal microbiome, our turf garters, and invading microbes. And those, the resident microbes, the mutualistic microbes, actually help us to antagonize invaders. So again, on this topic of biofilms, biofilms are super important in nature and in healthcare. We have biofilms that form on our bodies on a regular basis that you find on your teeth. That's why you have to brush your teeth, because bacteria stick and form these films, and then the films can calcify. I don't know that much about dentistry. So again, a lot of this formation and this synergy is due to bacteria's ability to do quorum sensing, to essentially talk to each other using molecules and let each other, and basically count how many other bacteria are around. And when there's enough of them, they can launch an attack. They can launch a community. And they can recruit other bacteria and even other organisms to their community. And it makes them very resistant to, removal. And so a lot of bacteria can kind of exist in two different modes. They can be either planktonic, meaning that they're free swimming, single-celled bacteria, or they are in a biofilm, where they're living in a community and growing together, and sometimes that means they're expressing different genes, they're making different proteins when they're in that kind of environment. And biofilms are particularly problematic because they're very resistant to antibiotics, because it's just difficult for antibiotics to penetrate them. And so infections that are caused by biofilms, particularly if they're internal, and these often happen on artificial body parts or prosthetics. So like if you have an artificial hip, or if you have like artificial heart valve, or you have some like pins or plates holding bones together, those are all surfaces that bacteria can be introduced to and grow on after they've been inserted. And then it can be really hard to treat the infections because they're too dense. And a lot of times treatment ends up being surgical. You have to go in and literally like brush off that thing just like you brush your teeth, to really get rid of that biofilm. And so in one of the TED Talks you watched about quorum sensing, they were even talking about using quorum sensing blockers to treat certain materials like plastics for catheters, to prevent biofilms from being able to form on them. Because that's the other issue, is biofilms can form on equipment, medical equipment, that then causes an infection. So catheters and ventilators are very problematic for causing infections because biofilms can form on them. So the way that biofilms become antibiotic resistant and contribute to the greater problem of antibiotic resistance is you have some planktonic free-living bacteria. They adhere to a substrate, to a surface material, and they start ramping up their glycocalyx production, and they all stick together, and then they're growing and growing and growing. And the cells in the center, they kind of turn off and go dormant a little bit. They're what we call metabolically inert. They're just not actively reproducing or metabolizing. And so they are more resistant to antibiotics, because even if the antibiotics penetrate that deeply, in order for antibiotics to work, the bacteria actually has to take it in. It's kind of like, think of an antibiotic as a poison, and you actually have to drink it in order for it to poison you. Well, these bacteria aren't drinking. They're just kind of sitting around, and they don't need to eat or drink. And so the poison doesn't affect them. When you have a whole bunch of cells growing and growing and growing, chances are you're going to get one that just gets a lucky mutation that makes it resistant to antibiotics. In fact, low level of antibiotic exposure actually selects for antibiotic resistance and encourages antibiotic resistance. And we're going to watch a really cool video of that in a later lecture. So it can actually promote antibiotic resistant formation. So biofilms are particularly bad for not just for the individual who has that infection and makes it very difficult to treat because it's not effective for antibiotics, but also by trying to chronically treat these with antibiotics, prolonged antibiotics, it can also lead to the development and evolution of new antibiotic resistant strains that then can be passed on through population. So biofilms are a pretty important issue. So how do bacteria grow and multiply? Well, pretty simply. They basically double all of their innards and then split down the middle. And that's called binary fission. So they double their DNA, they make a copy of their chromosome, they split their ribosomes and cytoplasm, and pull them to two different sides of the cell, and then some cytoskeleton proteins form a band in the center and pinch the two membranes down, and now you have two separate cells, a daughter cell and a parent cell. And they just keep doing that over and over again, binary fission. So bacteria have very, very short life cycles or very fast generation times. So a generation time is how long it takes for a bacteria population to double, and essentially how long it takes bacteria to divide. And, in our life, generation time is like, what, 15, 20, 30 years? And for bacteria, it's every 30 to 60 minutes. So, on planet Earth, there's maybe in a family, there's 5, at the most, probably 6 generations that could possibly be alive in one, life cycle. Whereas bacteria in, just their lifespan, if you were, okay, so how like in dog years, you say like, oh, dog, 1 human year or 1 dog year is 7 human years. So 1 bacteria year is probably like, I don't know, 100 human years or something. I'm just making up a large number, but bacteria live for a relatively short time, but they reproduce really, really quickly. That was a dumb analogy, but I tried. So essentially, after 24 hours, so if you think of this doubling time, right? So after 30 minutes, 1 cell becomes 2, and then after an hour, 2 cells become 4, and so on and so forth. All right, these early numbers are small, but when you're growing, when you're doubling like that, that's called logarithmic growth. And logarithmic growth quickly increases exponentially. It's exponential growth. And so over the earlier time frames, you still have relatively small amounts, but then it becomes significantly higher. Those numbers start to get really, really big. And so by 24 hours, you have millions of bacteria from one. One bacteria can turn into millions of bacteria. So they won't keep growing, though, to infinity, like that logarithmic chart on the previous slide suggested, because eventually they start using up all their resources and crowding each other and getting old and dying. So if we were to if we were to inoculate a culture and watch its growth, it goes through 4 different stages. The first stage is called the lag phase, and this is when the bacteria aren't even necessarily really dividing yet. They've just been introduced to this new media. They're, just metabolizing and going on their lives. And then they're like, okay, yeah, we're going to settle here. We're going to start growing our family. And they start dividing. And when they start dividing in earnest, the first couple of divisions are that doesn't make a big difference. But then it really does. And so that is called the exponential growth phase. And this is the phase where the bacteria are actively dividing and dividing and dividing and dividing generation after generation, right? But ultimately they reach a point where the population becomes overcrowded and they start running out of nutrients and space, and they also start getting kind of old for their little bacterial lifetimes, and they start to die. So the stationary phase is their sort of like, well, they're not growing anymore because they're a little too crowded and there's really not a lot of resources and nutrients to go around. And then they all just get old and die, and so there's a death phase. So those are the 4 phases. The exponential growth phase, by the way, is also sometimes called the log phase because it's logarithmic growth. The period of time, like when you're doing experiments with bacteria, you want them to be in this exponential growth phase. And like if we were in lab and looking at microbes under the microscope, I would be freshly inoculating culture the day before lab so that we could look at fresh, healthy cells that are in the prime of their lives in the exponential growth phase. All right. When they reach the stationary phase and death phase, they just kind of look crumbly. They don't, behave right or stain right, and it's just not an ideal time to view them. The other thing is their metabolism slows down as they get older, if you will. And remember that for things like antibiotics and stuff to work properly, the cells have to be actively taking in nutrients and metabolizing them. And so organisms like pathogens are also most susceptible to antibiotics in this stage of their growth rather than later stages. But these guys are weaker. So if we relate this to a pathogen causing an infection, it kind of follows the same sort of growth phase. And the difference is that they usually start dying off due to the immune response toward them. And so early in the infection stage, you might not be very symptomatic because there's not as many bacteria there. And then once there's exponential growth, you start getting symptoms. So especially in a lab setting, when you're trying to quantify the amount of bacteria in a culture, there's a couple of ways to do that. One way to do it is doing what's called a viable plate count, where you take your culture. So if this is a flask that you inoculated and you grew bacteria overnight in this big old flask, and you want to know, to be able to count how many cells are there, you take a little bit of that media and you do what's called a serial dilution. So you put a little bit of that media in a tube, and then you take a little bit of that media and add it to, maybe take 1 milliliter of this and add it to 10 milliliters of media, and then you take 1 milliliter of this, add it to 10 milliliters over here, and so on and so forth. So each solution is more and more dilute. I just paused because that doesn't make any. Oh, I see what it's saying. Okay, so this is it. In this picture, it's kind of backwards because in this picture, it's measuring. Okay, this picture is just measuring the growth after certain periods of time. So, like after 60 minutes of growth, you add some to here. After 2 hours of growth, after 3 hours of growth, and so on and so forth, and you can see that population rising. All right, you can also do this sort of in reverse with the serial dilutions, like I said, in order to count the number of bacteria in a culture by being able to count how many colonies grew from that volume that you use. So that's a viable plate count. Turbidity measurements use light to measure basically the cloudiness or turbidity of a solution. So remember, when you have a sterile growth media that's liquid, it's transparent, you can see through it, it looks kind of like apple juice. And when it has bacteria or microbes growing in it, it becomes cloudy. We say that the word for that is turbid. So this is a very turbid medium on the right here. So you can have something in between, something that's a little bit cloudy. And so what spectrophotometers do, or spectrometers do, is they send light through, they basically measure how much light can pass through. Oops. And so the more turbid it is, the less light can pass through, and the machine can calculate. There's formulas to calculate based on how much light passes through, how many cells that is. So that's another way that labs will use to identify really what stage of growth a culture is in order to get it in that exponential stage. Other ways that you can count the number of cells in a culture is through like physically counting them. So there are special slides that are, I'm blanking on, a cytometer. Cytometer is a slide that has a tiny little like graph on it. Like you can't see it with your naked eye, but you can see it under the microscope. And so you put a certain volume of culture on there, and then you can, when you do the microscope, there's like a grid, so you can count how many are in a certain grid space, and then there's a mathematical equation for multiplying, figuring out how much liquid is in each grid, and do some multiplication to figure out how many cells are in the volume, total volume of your culture. So we call that direct cell counting. You're directly counting the number of cells. And I did this a lot when I worked with human cells in college, actually after college, but before grad school, I worked with I was actually studying a virus. I was studying HPV, and we grew viruses in HeLa cells, human cells. And so I would have to count how many cells were in my culture, and I used a cytometer. So that's a manual counting with your eyes, and like a counter, that's me tick, 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 tick on a counter. A Coulter counter and a flow cytometer, these are big expensive machines that you basically put your culture in, and they use lasers to count the cells as they pass by. So it's a more automated cell counting technique using technology. Other ways to count cells, there's a type of genetic counting mechanism. It's called real-time PCR, where basically it counts, it's a way to quantify the amount of DNA or genetic material, and so that's another way to quantify bacteria in a sample. Oh, and that's the end of this lecture. It's a short one.